a huge fan of tryptophan. Why? It absorbs UV. This allows us to protein see. So the amino acid tryptophan is a key component of proteins as well as a precursor to some hormones like melatonin and serotonin. What happens is that tryptophan also has this special property called resonance or electron delocalization. Basically, it has those big rings that actually have this special property that's going to allow them to absorb UV light. And so this ultraviolet light, if we can measure the absorptions, we can measure the proteins kind of like stealing this light, then we can track proteins, say when we're purifying them, as well as be able to convert how much light was stolen into the amount of protein. So we can figure out protein concentration based on the absorbance of UV light. Typically we measure this at 280 nanometers, which brings us to having to talk about light and why light is absorbed by some molecules, well, by most molecules, but different wavelengths of light, and how the electron delocalization of tryptophan, as well as the other aromatic amino acids, helps it absorb this UV light, and how we can take advantage of that. So let's start with light. Let's go.
It's easy to take light for granted. I mean, it's just that thing that like shines out of light bulbs, right? We typically only recognize the importance of light when we notice its absence. And so maybe you turn off that light bulb, or maybe there's a molecule that's stealing light. And maybe it's not just stealing all of the light, but it's stealing a particular wavelength of light. And this is going to allow you to see color, or you can detect the stealing of that wavelength of light using a machine like a spectrophotometer. So in order to understand what's going on, we need to understand what light is. And so it's not just the thing that shines. It's actually made up of a lot of little things. And it's made up of these little particles, these little energy packets called photons. And these photons have these fixed energies, and they're traveling in waves. They're going up and down and up and down and up and down and up and down, these zigzags. And the more energy those different photons have, the faster the waves that they're going to go. And so when we talk about fast, though, speed of light is the same no matter what the wavelength is. So if those photons have more energy, they're going to go up and down more, but they're still going to travel at the same linear speed. So if you have a low energy photon, it's going to be going at a low frequency, so it's not going to go up and down as much, but it's still going to travel at the same like linear rate as something that is high energy. Something that's high energy is going to have a high frequency. It's going to go up and down a lot, but it's going to go at the same linear rate. So we have the same speed of light. If you're going up and down a lot more, though, you're going to have a shorter wavelength in order to squish more up and downs into the same amount of space. And so we can talk about the relationship between energy, frequency, and wavelength. There's this direct and like inverse relationship between, depending on which one you're looking at, but basically the higher the energy the photon has, remember those photons have fixed amounts of energy, different wavelengths are going to have different amounts of energy in those photons. And the reason why they have different wavelengths, if they have different energies, is because if you have a higher energy, you're going to go up and down more in order to travel kind of more distance over the same linear distance to get rid of that energy. Think of a little kid like running alongside grandpa, but they're walking at the same pace. So they're keeping track with one another, but the little kid's going zigzag, zigzag, zigzag a lot more. And so it's going to be having a higher frequency, the number of times it would like pass through a specific point. And so more energy, higher frequency, but it's going to have a shorter wavelength in order to squish all those up downs into the same amount of space. So when we talk about frequency and energy, these are going to be directly correlated. So higher energy, higher frequency, but that's going to be inversely correlated to the wavelength. So higher energy, higher frequency, but lower wavelength. So if you have a shorter wavelength, it's going to be higher energy. And if you have a longer wavelength, it's going to be lower energy. So for example, if you were to look at blue light, well, blue light's going to have a higher energy than red light. And blue light is therefore going to have a shorter wavelength. We typically talk about wavelengths in terms of nanometers, which are going to be a billionth of a meter. And if we look at red light, we see that it has a longer wavelength. And so it's going to have a lower frequency, a lower energy, but a longer wavelength. And if we go to even longer wavelengths, we go to lower energies, we get infrared. And if we go to shorter wavelengths, higher energies, we get ultraviolet. So you can see that the visible light spectrum is just a tiny little portion of what we call the electromagnetic radiation, or the electromagnetic spectrum. So all of this stuff is light, but we can only see a specific part of it. Because we can only see a specific part of it, we're not going to notice if part of the other, if like microwaves get stolen or if UV rays get stolen, unless we have a machine that can measure the UV. And so this is what we're going to see later, is that we have a machine that's going to allow us to see if UV light gets stolen. But normally we can only see if visible light gets stolen. And why light gets stolen is because different molecules can absorb different wavelengths of light. Now, a lot of times they're going to be absorbing wavelengths that are either too low energy or too high energy for us to see. So when we're talking about biochemical molecules, often these are involving the energy that they can absorb is going to be really high energy. And it's going to be so high energy that we can't recognize if it gets stolen. So even if it does get stolen, we're not going to be able to notice. What happens is that certain configurations of atoms, in particular, this thing that we'll talk about, this resonance or electron delocalization. So in the case of the aromatic amino acids, we'll see that this happens in a ring. We get this aromatic amino acid where we have this delocalization around the ring. And we'll talk much more about what this is representing. But for now, just know that in order to absorb light, you need to the molecule has to have a configuration that can absorb a specific energy, because those photons have specific energy. And so the specific energy of that photon has to match the amount of energy that that atom can absorb. And if that absorption is and because different wavelengths have different energies, different molecules are going to absorb different wavelengths of light. Now, though, if they're absorbing light in a range that we can't see, then it doesn't really matter unless we have some machine that we can detect it. If they absorb light in the range that we can see, well, now we see color, because what we see when we see color, white light is made up of all of that visible light spectrum. So this portion of the spectrum that we can see, if we were to take all of this light, we would see white light. Now, if you were to remove some of that light, well, now it wouldn't look white anymore. So if you take away some light from the visible spectrum, it doesn't look white anymore. And this is why we get colored compounds. Now, when you look at a colored compound, like a dye, often what you see is you see a lot of that resonance, of that delocalization, a lot of aromatic groups, and things like this. And this is because that resonance, as we'll discuss, lowers the cost for exciting those electrons. It lowers the amount of energy that's needed in those waves. So it lowers the amount of energy of the photons that are required in order to excite the molecule. And so if you are lowering the amount of energy, remember how I said that normally they absorb really high energy. Well, if you lower that, well, now you're getting into a range that we might be able to detect. And if you get low enough into the range where you're absorbing visible light, then we can see that. And then we can see colored compounds. And so if you have a lot of that conjugation, you're going to get something that looks like a different color. And depending on what wavelength it ends up stealing, you'll end up getting a different color. And so you can look at a color wheel, and basically things will look the color that that's opposite of the color that they stole from the color wheel. And so if something were to steal, say, yellow light, it would look all purpley. If something were to steal blue light, it would look orangey. But in the case of a protein, typically it's not absorbing enough of that light to actually be visible. It's not absorbing that low enough of energy of light, but it is absorbing in the UV range. And the UV range, well, we have detectors that we can use to detect ultraviolet light. And we can use these spectrophotometers, I can never remember which words, but this machine that's going to shine light through a sample containing a protein or another solution, and basically measure how much of that light gets stolen along the way. And then we can use this to convert the amount of light that was stolen into the concentration of the protein. And different proteins are going to have different kind of conversion units. And so you can use a software like XPazi-Protparam to figure out what the extinction coefficient is, basically how much light loss corresponds to how much protein, because the more protein you have, the more light's going to get stolen. And therefore you can use that relationship to calculate how much of the protein was in there based on how much of the light was stolen. And because different proteins are going to steal different amounts of light, because the proteins are going to have different compositions and different proportions of these amino acids, these aromatic amino acids. And why I'm talking about the aromatic amino acids is because these are going to have those resonance. They're going to have that electron delocalization that's going to allow them to absorb that UV light. And so different proteins are going to have different amounts of these. And the more they have of these, the more UV light that they're going to absorb. So with the same amount of protein, if you had more of these aromatic amino acids, you would absorb more light. And so you would get a higher signal, even though you had the same number of copies of proteins. And this is why you have to calculate this different extinction coefficient for different proteins. So I'm talking about the aromatic amino acids, but really the big player here is going to be tryptophan. And so you can see tryptophan, it's going to have it's going to have like a lot of resonance on a lot of different opportunities for this light to be absorbed and for the energy needed to be lowered. And so we call this group on tryptophan this indole ring, and where it's mainly going to be absorbing is UV 280. And so we can measure UV 280 in order to measure the protein concentration and then calculate the exact concentration for our protein. If we have a pure protein mixture, we can use the extinction coefficient, or we can kind of use an average if we have a mix of different proteins. We can kind of use like the average value to correspond to the UV trace. And this is really helpful. Being able to see UV is really helpful, for example, in not only for determining concentration, but also for kind of tracking proteins, such as when you're purifying them with a machine like an ACTA, when they're doing chromatography. And much more in chromatography and other posts, but basically you're sending a solution with proteins through these columns filled with beads, and then the beads are going to separate the proteins based on their properties. And so if you look at the absorption spectrum, however, of a protein, you're not going to see just like a pure peak. Instead, you kind of see a couple different peaks. What's going to happen is not only do you have the protein absorbing predominantly at 280 from those sides, from the aromatic rings, but you also get absorbance from the peptide backbone, because that peptide backbone of the protein, that also has resonance, that also has that electron delocalization. And that electron delocalization is going to lower the cost, lower the amount of the wavelengths that's needed in order to be absorbed by the protein, and therefore you're going to be able to detect it. But 280 is going to be like the highest absorbance for the protein, the highest like protein specific absorbance. So we've kind of been skirting around this issue of what's actually happening when light is being absorbed. So let's step back and talk about that in more detail. So basically, what happens when light is absorbed is that you're going to be exciting electrons. And so electrons are what atoms use in order to form bonds to one another. So different atoms are made up of subatomic particles, so these smaller parts. These are protons, which are positively charged, neutrons, which are neutral, and electrons, which are negatively charged. When atoms form bonds to one another, they do so by sharing pairs of electrons. Much more on this in other posts, especially yesterday's post on phenylalanine. But in addition to sharing electrons with the atom that's next to it, so like say this carbon to this carbon, sometimes what they can do is they can share electrons with more than just the neighboring atom. And we call this electron delocalization or resonance or conjugation, where you're sharing with not just the one next to you, but more of them. And this can happen when you have more than enough for a single bond, but not quite enough for a double bond. So when you share a pair of electrons, you have a single bond, and two pairs, you have a double bond. But what happens if you don't have enough for a double bond? But if all of these are in an orientation that allows it, then what can happen is that they can kind of share their extras, and so that they have partial double bonded character throughout multiple atoms. And when this happens in a ring, then we call something aromatic. And this is going to come into play with the fact that when you have this double bond, it's going to lower the amount of energy that's needed in order to excite those electrons. And similarly, if you have resonance, you're going to lower the amount of energy that's needed in order to excite the electrons. And in order to excite electrons, what I'm talking about is basically absorbing light or energy from another source. But in this case, we're going to be talking about absorbing light. And so if the amount of energy that's needed to excite that electron corresponds to the energy of the light, well, then you can get light be absorbed. And if you can measure that light being absorbed, well, then you're going to be able to measure how much of the molecule was present, or see that the molecule was present in the first place. And so because you have this electron delocalization, you have this resonance, this this conjugation throughout in these amino acids, you're going to be able to detect the absorption of UV light in the case of these molecules. And so that is the basics of how these actually, how that resonance is going to affect the absorption. It's going to lower the cost to absorb the energy and push it into a range where we can then detect it. So most of the time it would be in a really high energy range that we can't see. But if you were to lower it down with that resonance, then you can get it into this UV range, which we can detect with the detector. And the main one that you're going to be seeing is tryptophan. Protons and the neutrons hang out in this dense central core called the nucleus, and then the electrons are whizzing around it. We can never know exactly where the electrons are going to be, but we can kind of predict where they like to hang out. And we can represent where they like to hang out in like orbitals and different energy levels. So different electrons have different kind of like energy levels that it can occupy, and it's outside the scope of this post to try to explain it in detail. But we can kind of represent these different energy levels through these things called Jablonski diagrams, which are kind of like apartment floor plans. So you can imagine the electrons as living in these different energy levels, and they can travel between these energy levels, but only if they absorb energy that is the exact amount to take them from like one floor to the next floor. So it's like they can't just like be suspended up here in the air, but they can go between the floors. It's not if you were to absorb that exact wavelength, well, now that electron can get excited and it can travel to a different floor. Now, different molecules are going to have different floor plans. They're going to have different like ceiling heights. So it's going to cost different amounts of energy to get between different floors. In the case where we have conjugation, when we have that resonance stabilization, it's going to make the distance between those floors shorter. And this means that lower energy, longer wavelength light, can sometimes even visible light in the case of dyes, now that can be absorbed. And so it's lowering the cost to get to another floor. And so you're lowering the wavelength of the light that gets stolen. And therefore you might be able to change it into something that you can see. Other ways that you can see something sometimes is that if it gives off light when it falls back down, we call that fluorescence. But other times it can kind of just fizzle back down and give off heat or something like that, because you can't stay in an excited state for long. So we have different molecules, have different electronic floor plans, so they can absorb different amounts of energy, different wavelengths of light. But if you were to look at the absorbance of a molecule, you don't just see a single like peak. And so although we might measure a protein concentration at like a UV 280, we do that because that's like the most protein specific wavelength, but not because it's the only place that a protein absorbs light, and even around 280, there's going to be some wiggle room. And you have this wiggle room because those energy levels, there's also these like vibrational states within these energy levels. So you can think of kind of like standing on a bed or standing on a dresser or a table. You're going to be starting at different heights. I mean, you can also come down to different heights. And so there's going to be some wiggle room in the absorbance and that in the emission. So like, so there's going to be some wiggle room in like the absorbance. So what wavelengths of light it can absorb. There's going to be one main one corresponding to like these ground states and stuff, but there can also be these main states, but there can also be like these various vibrational levels giving you that wiggle room, so that if you were to take the spectrum, you would see you measure absorbance over a variety of wavelengths, you would see a spectrum where you're getting absorbance at multiple places. not just because you have multiple chromophores, so you have multiple places on that molecule that can absorb UV light. So say all those different amino acid side chains and all those different peptide backbones. But at each of those sites, you also have some wiggle room. And so you get broader peaks in addition, as opposed to just like a sharp line. But we typically measure at a single point because then that gives us a way that we can convert easily to, say, concentration. But when we do so, we need to keep it in mind that different proteins are going to have different amounts of those different groups that absorb different UV light, which is why if you know the makeup of the protein, then the best thing to do is to actually calculate the exact extinction coefficient. So how much of the protein corresponds to how much light being stolen. And typically we're measuring at a UV 280 because this is going to be where we see the highest signal that's specific to proteins. So tryptophan helps us kind of like see proteins in this way. It also kind of helps us see proteins when we're doing X-ray crystallography. So this is a technique I talk about a lot more in other posts, but basically you shine X-rays at a crystal made of proteins. And those X-rays are going to like diffract. They kind of like hit the protein and they like go poof, like, like when you hit, drop a golf ball in water, those like ripples, that's what happens to these X-rays. And then those ripples interact with one another and they create this pattern of spots on a detector, and then you work backwards from that spots to figure out where the electrons were. And so because the electrons are going to be like centered around the atoms, like the core of those atoms, you can then figure out the structure of the proteins. And sometimes it can be really hard to orient yourself in the molecule and try to figure out what's what in just like that like electron density mesh that you get. But tryptophan, because it's big and it's got all these electrons to absorb, you often get a clear signal from tryptophan, and so it can help you kind of like figure out where things are. So that's just a cool side note. So tryptophan is an aromatic amino acid. Similarly to phenylalanine and tyrosine, which we looked at in the last couple days. When we look at tryptophan, it's going to be bigger and bulkier. So it's going to have a little more restricted movement. So when we talk about like hydrophobicity, so basically these different electrons, so these atoms are sharing electrons to form bonds. And if they don't share fairly, then you can get a partial separation of charge we call polarity. Now, water is very polar. And so it's going to, and it likes other polar things because they have partial charges. And so if something is polar, it's going to typically hang out better with water. We can think about things as like polar versus non-polar, but really it's normally a spectrum. And so in the case of phenylalanine, tryptophan, tyrosine is going to be the most polar, but it's still going to be relatively hydrophobic, so water avoided, because it has a lot of non-polar character. So carbon and hydrogen, they're going to share really fairly, and so you don't have polarity. But an oxygen, that's going to hog the electrons, making it partly negative, and the things it's attached to partly positive. So you get this polarity with tyrosine, but it's still going to be predominantly hydrophobic. With phenylalanine, you don't have anything that's offering polarity, and so it's going to be very hydrophobic. And with tryptophan, you're kind of in the middle. You're still very hydrophobic because you've got a lot of that nonpolar character, but you have a nitrogen, which offers a bit of polarity. But all three of these are going to be classified as aromatic because they have that situation where they have their electrons shared not only between the neighboring carbon atoms, but also between like all of these carbons in these rings, or all this nitrogen as well. And so all of this, you get this delocalized electron density above and below the rings, kind of like a donut. So that's where we can see tryptophan like in the lab, but where will we see it in our body? And so in our bodies, we see it used to make serotonin and melatonin. And so these are chemical signals, signaling molecules that our body uses in order to communicate. And so I talk much more about this in my post on like tryptophan hormones and the turkey make you tired myth. But basically, serotonin is going to be made, but basically from tryptophan, you can convert it into serotonin, which is often associated with like happiness. This is also known as 5-hydroxytryptamine or 5-HT. And then this can get converted into melatonin, which is associated with like sleepiness. Serotonin is used as a neurotransmitter, which means that it gets sent from a brain cell, from a neuron, to a neighboring cell. And so it's going to be sent these really short distances from a neuron to another cell. Melatonin, this is going to be a hormone. And so it's made in the pineal gland. And when you see gland, think hormone. A hormone is going to be a chemical messenger that's actually secreted into the bloodstream. And if something's secreted into the bloodstream, now it's not constricted to moving in between short little distances. Now it can travel throughout the body, and different cells, if they have the receptors for it, they can respond to the signal. They can bind to that molecule and then like change shape or set off a signaling cascade, etc. That allows the cells to respond, and the cells will respond in different ways depending on the cell type and the receptor type and things like this. But you can make both of these things from tryptophan. And they can have this synthesis pathway that kind of looks a lot like the synthesis pathway that we saw, or at least somewhat like the synthesis pathway that we saw yesterday when we were talking about tyrosine and how we can make the catecholamine hormones and neurotransmitters, so adrenaline and noradrenaline and dopamine from the amino acid tyrosine. When we looked at that pathway, we saw that we had a bunch of different enzymes, so a bunch of different reaction helpers, and that they were using helpers, these helper molecules, cofactors, which are basically like basically small molecules and things that these enzymes can bind to in order to help them do their job. And so we're going to see some of these same cofactors when we talk about how the tryptophan hormones are made. But when we're talking about the tryptophan hormones, here we're going to be converting it into different molecules because we're starting with a different molecule. But you can see we even use some of the same enzymes. So this dopa decarboxylase, well, you might remember if you watched yesterday's post, this is also referred to as like amino acid or aromatic amino acid decarboxylase because it can be used to decarboxylate different aromatic amino acids. And so it can be used to decarboxylate hydroxytryptophan, which was made from tryptophan. And this is that THB that we saw yesterday and that POP that we saw yesterday. But basically, you can see that to get from tryptophan, you can add an oxygen, get 5-hydroxytryptophan, then use that dopa decarboxylase to remove that carboxyl group. And this is going to give you serotonin. And then you can further modify that serotonin, add this acetyl group, and then add a methyl group. And so this is the same, this is similar to what we saw yesterday, how we could use methionine. We can use SAM, which takes a methyl group from methionine and transfer in, or it's a cofactor that allows an enzyme to take a methyl group from methionine and transfer it onto a different molecule. In this case, it's transferring it onto N-acetylserotonin to give you melatonin. And this is only going to happen in specific locations. It's going to happen in a region of your brain called the pineal gland. And this gland, remember, gland is telling you hormone, is going to do this transfer. Tryptophan gets a bit of like a VIP deal when it comes to getting into the brain to be used to make hormones and neurotransmitters. So basically, tryptophan throughout the bloodstream, it's kind of bound to a protein called albumin. It's this really abundant protein in our bloodstream. And it has to, so tryptophan has to compete with other amino acids, these like large neutral amino acids, for a lot of the same receptors and importers to get into cells. But what happens is that in your bloodstream, the tryptophan kind of gets tied up because it's bound to that albumin. Less of it's going to get into those other cells. But you're still going to have tryptophan left over when you get to the brain, therefore, because you haven't used it all up, because it's been bound to that albumin. And when you get to the brain, well, now here you don't have as much competition because you've imported a lot of those other amino acids, especially if you have like insulin secreted, which is going to, when you eat carbs, then your insulin secreted, and it tells your body not just to take in glucose, the cells to take in glucose, but also to take in those the amino acids. But because tryptophan's tied up, it can't be taken in as much. This is going to leave more available then to get into your brain. And when it gets to those brain receptors, well, here they're going to have a higher affinity for that tryptophan as well. So in this way, your brain is still able to get plenty of the tryptophan that it needs. But a note, the melatonin that's getting made in the pineal gland, which isn't protected by that blood-brain barrier. So under stress conditions, tryptophan can be broken down by this like kynurenine pathway that breaks it So under stress conditions, like stress hormones can cause tryptophan to be broken down by this kynurenine pathway. And so it's going to build different molecules that can be used to do things like make NAD plus and alanine, as well as some other molecules. And so more on that in posts and stuff, but I'm not going to get into that here. Just know that this is not the only pathway that tryptophan can take. Speaking of pathways that tryptophan can take, let's talk about how we can break it down. Speaking of pathways that tryptophan can take, let's talk about how we can break it down. So tryptophan is considered glucogenic and ketogenic. It can be converted into acetyl-acetyl-CoA or acetyl-CoA. These are both ketogenic. They can be used to make ketone bodies or lipids, much more in this in my leucine post. It can also be converted into pyruvate, which can be converted into oxaloacetate, which can be used to make glucose. So it's considered glucogenic as well. So it's both ketogenic and glucogenic. But it's also essential because we need to get it pre-made from our food. We can't make it ourselves in our body. So tryptophan is really helpful for helping us see proteins, as well as for allowing our body cells to communicate with one another. So through serotonin and melatonin. And it's also just really cool, I guess. It has like a it has the most complex of the metabolic pathways and things. So I'm not going to get into all of it, but no. So this is tryptophan. It's aromatic. It is mostly hydrophobic, but it's got a little bit of polarity thanks to that nitrogen, so it's not quite as bad as phenylalanine, even though it's a lot bigger and more carbohydrate-y. But these are all aromatic. Because this is tryptophan is like kind of like the most aromatic, what's going to happen is that it's going to absorb the most UV light, and so it's going to really help you see proteins when you're measuring the absorption. It can also be used to make hormones and neurotransmitters, so it can be used to make serotonin and melatonin. It has a very complicated breakdown pathways, the most complicated of all the amino acids. We can't make it in ourselves from our body, so it's considered dietarily essential, meaning we need to get it pre-made through our food. And then our body uses it in a variety of different ways, not just to incorporate into proteins, but also to do other things. And so I hope that helps you understand tryptophan and light absorption and all that stuff.